Today, Natalie Graham, a graduate student with Rosemary Gillespie, who is graduating this summer, will be presenting her research on dynamics during community assembly, evaluated using DNA metabarcoding of arthropods on islands. Her research is being done through a permit with the HETF. Please note that permits are required when entering the HETF for research purposes. Natalie, you can start whenever you're ready.
I'm just trying to set up the Zoomy with my, let me try to reshare again. Okay, perfect. And everyone can see my screen?
It's all good.
All right. So as Olivia said, I am sharing some of my dissertation research with you today. So I'm excited about that. I'm excited to be graduating in just over a month. So I am a graduate student with Dr. Rosemary Gillespie in the Department of Environmental Science, Policy and Management at the University of California, Berkeley. And I've been fortunate enough to be working in Hawaii for my dissertation research. So I'm interested in understanding biodiversity dynamics and how those change through time. And I've evaluated this using some novel high throughput DNA sequencing methods, so specifically DNA metabarcoding of arthropod communities on islands. So the primary question I'm interested in asking is, how are communities changing through time? So if we go from primary substrate like this bare lava, what do communities look like as they go to like this younger community, like it's about 50 years old, to something like an evolutionary stable community on the island of Kauai? Biodiversity at any point in time, we know it's going to be governed by local and regional processes, but it's also going to be governed by these large scale historic biogeographic processes. So unfortunately, most studies of community assembly have focused on short-term dynamics because it's difficult to study how biodiversity is changing over longer-term evolutionary timescales. What this means is that like the explicit link between macroecological and macroevolutionary patterns and processes on the formation of biodiversity has been mostly unexplored. So here we're using communities of arthropods and plants to study how communities change through time. So when I'm talking about biodiversity, we typically think of that as like the level of like what are different species in a community. And for those of you that are uninitiated about what an arthropod is, arthropods are include insects, but they also include kind of insects in their kin. So spiders, mites, centipedes, millipedes, amphipods. Some of you might think of these as just being all of the creepy crawlies. So that's kind of species biodiversity at the species level. But we also have biodiversity at the genetic level, as well as at the ecosystem level. And so I'd like to show you how that changes through time at all of these different scales. And we know that biodiversity is shaped by interactions of organisms, both interactions with their environment, also interactions with one another in the community. And we know that the relative importance of these abiotic and biotic interactions is also going to change through time. Interestingly, models have been developed to infer this dynamic process, but empirical data is almost entirely lacking. And I mean, the reason for this is, how do you sample a whole community, right? And recently, the technology has been developed to do just that. So to sample whole communities in kind of one fell swoop using high throughput DNA sequencing, and specifically here, DNA metabarcoding. Of course, not only are we trying to sample whole communities, we're also trying to track them in a very changing world. So the Anthropocene offers these added challenges to understand biodiversity baselines, community stability, resilience, and change. But besides these challenges, it also makes it imperative that we find rapid and efficient solutions to understanding biodiversity and how it changes. So isolated islands provide this wonderful simplified system to study communities. Multiple islands of different age can actually serve as ecological and evolutionary time series. So the process of colonization, where new species are arriving on the island, biotic interactions, where they begin to interact with one another through predator-prey relationships or herbivore plant relationships, parasitoid host relationships, and so on. Those are going to be occurring. And with enough time, you'll also have speciation, or the generation of new species. And then the process of extinction, when species are either become locally or globally extinct. And of course, there's going to be some sort of interplay between these ecological and evolutionary processes and community formation as well. So sort of a traditional approach to understanding community assembly is this community ecology approach. This is the idea of like a snapshot into ecological processes. So like looking at like what are the extent of where species live or their species ranges, how many species live in one area, what's the turnover between different areas, different species, and so on. Another approach might take into consideration evolutionary history, but historically this has been done for one lineage at a time. And in this case, you would be trying to understand what the genetic diversity is, how much gene flow is there, how species are migrating between different populations. Perhaps there's isolation between different localities, and that's leading to processes like genetic drift and so on. And what we've attempted to do is to sort of take these snapshots of community assembly and turn them really into a moving picture by doing genomic sampling of whole communities across a time series. And so really this is the first like large scale empirical data set to test theory. And so we're excited about the ability for being able to test community assembly processes over extended space and time. So for today's talk, I'm going to first explain the study design, how we were kind of able to isolate the effect of time. I'd like to explore some of the benefits and challenges of molecular biodiversity assessment. And then I'd like to explain to you how biodiversity at these many different scales, this genetic scale, the species scale, and this ecosystem scale, is dynamic during community assembly. And then I'll highlight for you the value of this methodology for understanding and predicting community stability. So our first stop along the way is explaining the study design. So the Hawaiian archipelago, because of the island chronology, is this ideal system for evaluating community assembly. As with other hotspot archipelagos, the volcanic origin of the islands results in a stepping stone-like geographic arrangement as the Pacific plate moves across the volcanic hotspot. So this geologic history is actually mirrored in the pattern of evolution for many Hawaiian lineages. Thus, there's this tendency for many lineages to progress from older to younger islands. This is known as the progression rule. What this means is that multiple lineages are establishing and assembling, interacting and adapting over a similar time frame that plays out over extended time. So here what we did was we leveraged the power of high throughput sequencing, this DNA metabarcoding method, to examine the role of extended time and space in dictating community assembly patterns. So we sampled at 14 different sites on 4 different islands. We sampled a substrate age that ranges from 48 years down to 4 million years. And we tried to hold elevation, temperature, precipitation constant as much as possible. This is because we were really interested in just looking at the effect of time. So importantly, what this means is that each community represents a different stage of development. So we used a variety of different collection methods. In the bottom left picture and in the first video is a beat sampling method. This is where arthropods are aspirated off of a beating sheet after they've been shaken from a particular plant species. We also used a tree climbing method where we climbed the tree and then we cut particular branches off, and we took that branch back to the lab, and we were able to remove the arthropods from there. We also used some passive trap methods. So we hung malaise traps, which are basically like mosquito netting tents. And we put ground malaise traps up. That's the picture in the bottom left. We also sampled the leaf litter. So that top right picture is a malaise trap, and the arthropods are sampled from the soil there. So we kind of got every like level of the stratified forest. Today, the data I'm going to be sharing with you is from the beat sampling method. And I think this is the most exciting method because you are able to capture the relationship of the plants and their arthropod associations. What we did to make this quantitative is that when we went into a site, we looked at how much the relative abundance was of each of the plants, and then we sampled according to that relative abundance. So if there was a whole bunch of this tree fern, then we would sample that in larger proportion. Then we used this high throughput DNA sequencing, or DNA metabarcoding. And for this method, you actually collect all of the samples into a single container, and then you process them from DNA extraction all the way through to a DNA analysis as this one single pool of samples. To reduce the bias that might be introduced from processing all these samples together, we actually sorted them by size. We picked the cytochrome oxidase 1 locus, which is just a particular section of DNA, because there's actually a pretty good reference library. So samples of known specimens have been sequenced for that same section of DNA. And so we can relate our DNA sample back. So that's essentially what metabarcoding is this idea of taking a whole sample of arthropods and extracting the DNA of that whole bulk sample all in one container, and then amplifying just one section of that DNA, in this case, the cytochrome oxidase 1 locus, and then sequencing all of that. And so it's very important to understand that each individual arthropod, you get an individual sequence back from that. And I'll have a little diagram to kind of explain that in another slide. So to just kind of overview the study design, we had these 14 different sites. At each site, we had 6 different replicates. There were any number of plants at each site, and that varied across the archipelago. And then for each plant, we sorted into these different size categories just to help kind of reduce the bias in the sequencing process. And then we get out lots and lots and lots of DNA. So I want to explain a little bit about the benefits and challenges of this molecular biodiversity assessment. We know that community ecology is going to really rely on accurate assessment of species richness and abundances, and that can be a lengthy data collection process. You need to identify all the species, count how many individuals of each one that there are. So it's interesting that and fortunate that we can actually approximate this using molecular methods. Something else is the idea of cryptic diversity, so kind of diversity that's below the species level. Often that wouldn't be detected with traditional methods, but we can actually get at that with molecular methods. We get this genetic level of diversity. We need absolutely to be able to separate out native and introduced taxa. How do you do that if you're not, holding a pinned specimen in your hand? And then also getting at species interaction. So, typically you would observe a species interacting in nature for a while, and that can be time consuming. So how might we get at that with molecular biodiversity assessment? So looking at our first challenge, we're thinking about alpha diversity. Really what we have for DNA metabarcoding is a whole bunch of different sequencing reads that come back. Remember, it's the whole community that's being sequenced at the same time. So all of these reads represent the actual DNA sequence of that individual arthropod. And there might be a whole bunch of reads that are representing this one, what's called operational taxonomic unit. So the first thing that we do is we kind of group these reads by similarity of their DNA sequence together into something that's more of a biologically relevant unit. And that might equate one-to-one with what a species is, or it may be that there's this level of genetic diversity where there's actually a couple different OTUs that relate to one species. So there's kind of population level differences. And what we then do is we want to identify what those are. So we search them against a reference database. Interestingly, even though the cytochrome oxidase 1 locus is really useful and has a pretty large number of sequences in it, it's not complete, especially for tropical areas and hyperdiverse taxa like Hawaii. So we actually use DNA from voucher specimens and collections to improve on the reference library and make it easier to identify species. Okay, so another thing is getting at abundances. How do you do that again if you're not counting the number of individuals in the pool? And it turns out that there's a relationship between the number of taxa, or the proportion of taxa in a community, and the proportion of reads that you recover. And it's a nice, reliable kind of relationship. The slope, or how many reads you get out of how many taxa that you put in, might be different between like a spider and a bee, for example. But work was done in our lab by a postdoc when he was here, but he's now a professor in the University of Trier in Germany, Henrik Krenwinkel, to kind of be able to reliably compare the abundances between sites. So we can say confidently that we can capture abundances, which is important. You do have to be careful that the section of DNA that you choose is going to capture your community of interest. So in our case, the cytochrome oxidase 1 locus. So getting at the idea of cryptic diversity, here, this picture on the left, there's actually 16 different species there, but they look very similar. And so, with DNA, you can actually distinguish between these species that look similar. And you can also distinguish the diversity within a species. So this is like haplotype network. Essentially, it's just representing the frequency of a gene, as well as like what the variation is. In each of those steps is like the number of base pairs or DNA sequence differences that there are between different groups. So we get this genetic signature of whole communities, which is important. So again, getting at the challenge of like, how do you separate the native and invasive taxa? Well, it turns out that there's this wonderful signature in the DNA itself for native and invasive taxa. So this is a phylogeny. This represents all of the species that we sampled in our whole community, and they're color coded by order. So for example, up here in the gold, you see the spiders. There's a signature of the differences in native versus introduced taxa. And it turns out that native species, because they have evolved on the islands, they're very closely related to their sister species. And so they tend to have short branches, basically not very many base pair differences between them and their nearest neighbor. It's the inverse with non-native species. They haven't evolved. They don't have any real close relatives on the islands, and so they tend to have these really long branches or large amount of differences between them and any other relative that might be there. And so we really need to be able to, separate these in order to answer questions about ecology and evolution. And so in collaboration with Jeremy Anderson and a master's student of his, Jackson Callahan, we developed this machine learning application for separating native and non-native taxa. And I would be happy to address any more questions about this, but just the idea is that it uses a specific algorithm called random forests in a package sklearn that's implemented in Python. And it compares those features of the DNA, basically those short and long branches. And if you're able to identify some species, and you can name whether they're introduced or endemic, it can kind of fill in the blanks for the ones that you don't know and identify them as endemic or introduced based on those sequence characteristics. So please ask me more if you're interested in that, and I'm happy to share the link to the package if anyone is interested in trying that out. How do we get at species interactions? That's an interesting question, right? Because you're not actually observing the species in nature. And so let's just think a little bit about what our hypothesis would be for species interactions across Hawaii. We might expect a pretty simple network in the younger communities, so like on the Big Island of Hawaii, just to orient you a little bit about what an ecological network looks like. In this case, we're just looking between a relationship between species pairs. So this would be like a predator on the top and a prey on the bottom, or an herbivore on the top and a plant on the bottom. So just, one-to-one relationship. And a simple network would have a fewer number of species. The species are represented by the turquoise balls or nodes, and then the lines between them. And the thicker that the line is, the more often that those two species interact. So a more complex network would then have more species and more interactions. And an invaded network would perhaps have more asymmetric interactions. So where you have these a few species here on the right of this invaded network that are really having a lot of interactions with some other species on the bottom. And so we know that these take a time to observe in nature, but we can actually get these associations from the vegetation beat sampling that we did. If you're interested in getting trophic interactions, you can actually use molecular methods for this as well. And this is also some work that was done in our lab previously by Dr. Krenwinkel. So let's jump into some results now to look at how biodiversity across these different scales is dynamic. And before I give you results, I'm going to review with you a little bit about community assembly processes. So in any community, you have your actual observed community that you are sampling, which is some measure of a reliable sample of what the local species pool is. One nice thing about using this high throughput sequencing method is that we have a pretty comprehensive sampling of individuals in a community, because we're able to identify them regardless of if they're in like a different life stage. So if you think about how an arthropod larvae or like a caterpillar, for example, turns into a butterfly, it might be difficult to identify that caterpillar or that larval form. But if you're using DNA, you can get at that. So we have a pretty comprehensive sample of our local species pool. And that local species pool is interacting with a regional species pool. The regional species pool is just the greater area, so not in that specific locale. And it represents, other portions of the community that are interacting, maybe across a mountain range, so they're not tightly linked. And we can also think about the global species pool. This is really important, especially in the Anthropocene, where humans are moving a lot of species around. So we know that there's going to be some sort of neutral processes that are acting. You can think of neutral processes or things that are like not relevant to the biology of the organism itself. And then we also have abiotic factors and biotic factors or ecological interactions. And these are acting on both the regional pool and the local pool. And we know also then that these evolutionary processes are going to be happening as well. So the idea of new species being added or species adapting, developing new traits, and that the change in those species traits is going to affect the way that they interact with their environment. It's also going to affect the way that they interact with one another. So this is kind of that feedback part that I was talking about earlier. So let's think a little bit about biodiversity across scales now, and let's focus first on this level of species scale. So our first kind of question is, do the number of species increase linearly with time? So, we might expect that species are going to be added in these younger communities by the process of colonization to the island. They might also then be added later by the process of speciation, so literally the formation of new species. So we would expect that, from the youngest to the oldest island, there should be an increasing number of species over time. Will it happen linearly? I don't know. And that's part of the question. So when we go back to kind of our sampling design here, we see that we're sampling these whole communities over time. So, as I outlined, each of these different sampling sites, we had 14 different sites. Within that site, we had, these smaller white circles, which are the replicates. And so each of those replicates is kind of like a subsample of the community, and we can actually look at whether each of those replicates contains the same species or does it contain different ones, and that would kind of get at like the turnover or whether those species are actually on top of each other or not. So just sort of to expand on this a little bit more, we know that organisms are going to diversify in different ways. So we can look at these sort of 6 hypothetical different communities. We know that we might find sister species, in this case represented, for example, a single species by the green triangle. So we may find them all, these sister species all in the same community together, co-occurring within a geographic site. Alternatively, we might have these non-overlapping distributions of sister species. So here the closest relatives are not found in the same community. They're actually distributed across geographic space, or they're dividing up the geographic space. And they instead are found with things that are not their closest relative. So in Hawaii, we have some great examples of kind of either way of existing. So for sister species occurring within a geographic site, we have the adaptive radiation of Tetragnathus spiders. Here, there's been niche diversification and overlapping species, specifically with the spiny leg clade of Tetragnathus spiders. You'll find just one species on the younger island, and then 4 different overlapping species of different ecomorphs on the older islands. So kind of alternatively, contrastingly, we have the Lapala crickets. And with Lapala, they have few ecologically distinguishable features. So they're not adaptively radiating. They're actually not what we call non-adaptively radiating. And most closely related species are not co-occurring. And so we could say that like at our first site, there's going to be, at our youngest site, there's going to be this strong environmental filter. But as communities get older, the importance of species interactions is going to actually increase. And so we can kind of generate some hypotheses about what we might detect in these changes of diversity over time. So the first idea is that we can detect kind of a rapid accumulation in diversity over time. We would expect this to be associated with ecological affinities or different niche characteristics. So we can kind of think of this as filling the niche space or like the character space of different organisms, and it should increase steadily or rapidly. In the other example, where we have non-overlapping distributions of sister species, we would not expect to be able to detect these changes in diversity through time. And this is where we would think of more of this non-adaptive, more division of geographic space, where richness would increase more gradually or just be difficult to detect at the younger site because they're basically all the same. As you get to the older site, they've divided up the geographic space, and so you can actually detect those differences within a site between these different site replicates. So let's look at some results now. So here we have our example of richness accumulation, either with sister species co-occurring or non-overlapping distribution. So pulling out the spider example. And just to orient you a little bit about what these graphs represent, these are regressions of the number of ZOTUs, or the richness in the community, by log substrate age, or the age of the community. And as predicted, what we see here is when species are co-occurring, we see this accumulation of diversity over time. With the spider lineages, it's very, very distinct. And also, as predicted, we see that there's really no detection of diversity accumulation over time with the Lapala crickets and with other Orthoptera. And so that is really agreeing with our expectations for species co-occurring versus this non-overlapping distributions. Of course, with metabarcoding data, we can investigate this for multiple lineages. And so here we've regressed the richness by community age for every different arthropod order. And what we find is that the predictions for some lineages match this co-occurrence hypothesis. So I've kind of pulled out spiders, beetles, flies, true bugs, and the butterflies and moths. And they all have this pattern of increasing richness over time that is statistically significant. And interestingly, all of those lineages all share these commonalities, or this kind of co-occurrence hypothesis, where intra-island radiation is associated with these shifts in ecological affinity, this filling of niche space. And we also see that these lineages tend to, as they move from island to island, they follow that progression rule where they, hop down the island chain. Richness is accumulating steadily, and diversity tends to be lowest on the youngest island and then peak on the mid-aged islands or the older islands. Contrastingly, kind of along the lines of what Lapala, the crickets, the non-overlapping lineages, are Columbella, Orthoptera. So again, the crickets, Columbella are springtails, so they're not insects. They're kind of at the base of insects, and they're detritivores, and they're very important and abundant in the community. And then Socophora, which are woodlice. So these species are following this non-overlapping hypotheses, and they have some commonalities as well. Here we tend to see intra-island radiation be non-adaptive. Again, it's not that they're filling the niche space. It's more that they're just dividing up the geographic space in one way or another. This inter-island colonization, there's multiple inter-island colonization events, or they're going from the younger island back to the older island, so they're not following that progression rule. And richness tends to accumulate very gradually or not at all over time. And diversity is actually highest on the youngest island for a lot of these lineages. So interestingly, there's kind of insufficient information on the level of co-occurrence to predict for some of these lineages, just because they're understudied and there hasn't been much published about them in the literature. So we can't go in and say if they fit this co-occurrence hypothesis or this non-overlapping distribution hypothesis. But the data is pretty clear about what the relationship is for this richness accumulation over time. So in this case, we see that Myriapoda, which is the centipedes and millipedes, are following the richness accumulating over time, co-occurrence hypothesis, and same for Neuroptera, while the Malacostraca, which is your amphipods and isopods, do not. They're non-overlapping. Okay, so that was sort of lineage specific, but now we want to think about, like, if we put the whole community together, do we, as communities get older, do they become more diverse and or less homogeneous as a whole? And we might expect that those younger communities are kind of similar to one another with regard to within a particular site, that they probably share a lot of species because the younger communities are this harsh environmental filter, and there's just only certain species that can persist there. also, colonization has just happened because it's basically bare lava. And over time, we know that both the accumulation of niche differences between species is generating speciation, or this division of geographic space is creating new species at this non-adaptive process. And so we would expect that the communities would just be more diverse in the later stages. And so when we plot again by log substrate age or community age, and in this case, we're plotting on the y-axis this Sorensen similarity index, we see that, yes, as predicted, we get this fewer species that are shared among plots at older sites. So we see a statistically significant change in the diversity of the community over time. So within a site, the communities are becoming more diverse, which is a very interesting finding. And so now we're going to switch to thinking about diversity at the genetic level. And so the next question, our third question is, will there be a detectable change in the genetic relatedness of whole communities over time? And what my hypothesis was is that genetic relatedness would decrease over time. Now, why would genetic relatedness decrease? Well, at the younger community, species are colonizing from all over the place, mostly from the closer islands or different parts on the big island of Hawaii. But they're colonizing from other communities, and they're kind of a random assortment of the things that are colonizing from, remember, this regional species pool. And because this is this random assortment, they're not closely related to one another. So their genetic relatedness shouldn't be very, very high. And then we would expect then that over time, species should become more closely related. So just to sort of outline this hypothesis a little bit better, at the younger site, it's very ecologically open. There's little competition. Things are just arriving from the regional species pools. So we should have these long branches. Again, this gets at that signature of the community through the DNA. And so communities mostly made up of species that have evolved elsewhere. As you get to these next youngest communities, we would imagine that competition, and it's been demonstrated that competition becomes more important and more common. And in that case, species are going to start being pushed out of the community. So we would then expect there to be like not really a change in the branch length. The relatedness hasn't really changed, but the community is still made up of species that evolved elsewhere, but we've lost some branches. Finally, at the older age communities, there's been more time for evolution to take place. And so here we're seeing those really short branches. So now these are new species that have been added through the process of speciation. And the sister taxa are all really closely related to each other. So we should see a proliferation of branches where each branch represents a new species. And those branches should be short because those species have just evolved recently from one another. Remember, branch length is just associated with the amount of DNA that you share. So the shorter the branch, the fewer changes there are in the DNA between individuals. So let's see what we can do to test this. So what we did was we tested the distance between those individuals out at the tip of what we call a phylogeny, or the relationship of species in a community. And what we find is that this agrees with our hypothesis that the genetic relatedness changes over time. And it's a statistically significant trend. We also find that it's related to the number of taxa in the community, which is also increasing over time. So in this plot, we have, again, on the x-axis, the log substrate age, or the age of the community. And then in the y-axis, we have this measure of the mean nearest neighbor distance, or again, that distance of organisms at the tip of the tree. And so we see that, in the very youngest community, they really are not closely related to one another at all. The distance is very different, high between them. And then as we get into the older communities on the left-hand side of the plot in turquoise, those are the older islands where there's been enough time for evolution to take place. And in that case, you're starting to see those shorter branches develop on the tree. So as predicted, genetic distances are largest in the younger communities and decrease over time. So now let's look at the ecosystem level, and what we expect here is associations in the arthropods and their plant hosts to change over time. So this is kind of a very generalized hypothesis where I'm saying that network metrics are going to change over time. What are network metrics? Well, that's those relationships between the species on the top, the predator and the prey, versus the bottom, and so on. So let's look at some examples of networks and how we might measure them. So here, younger communities, like this community at Tree Planting Road, which is just 133 years old, we can look at the relationship between the community like this in a matrix form. Metrosideros polymorpha is the dominant tree species in Hawaiian rainforest, and you can see that by the darkness of the filled-in block here, that columbella are most highly associated with Metrosideros polymorpha. Kind of at the other end of the spectrum, vaccinium is another plant. It has very few or no myriapoda or centipedes and millipedes associated with it. Another way to visualize that is again using these bipartite networks. So in this case, again, the node or the species is represented by the blocks or the rectangles at the top. And this will be the higher level. So in this case, it's the arthropods on the top, and then the plants that they're associated with on the bottom. And the strength of that interaction is, again, the weight of the line. So like we saw in the matrix form, the columbella are most associated, have the most number of associations with Metrosideros polymorpha, and then with Dichronopsis, which is a type of fern, the Oluhe fern. As communities get older, we would expect them to be more complex. So this is a community at Molokai, which is 1.4 million years old. A larger number of species, first of all, is what jumps out when we look at the matrix. And then when we look at this bipartite network, what we see is that, yes, in fact, there is like a greater diversity and also a more one-to-one relationship of the network. So when I talked about network metrics changing predictably over time, I'm going to run you through a bunch of different metrics that we can calculate using these relationships of arthropods and their associated plants over time. So the first one is connectance, and kind of the take-home message from this is that there's a greater representation of generalist species at the youngest sites. Generalist species are species that can interact with multiple other individuals and be associated with, in this case, arthropods that are associated with multiple different plants. So this is defined as the fraction of all possible links or connections or associations that are realized in any network. And so here, again, plotting against law of substrate age or community age, we see that connectance decreases over time. Interaction evenness is the take-home message here, is that we actually see more balanced distribution of interactions on the older sites. So kind of a picture for you here, a less even network would be one like on the left that's more asymmetrical. A more even network is one on the right that's got this more one-to-one relationship. And so this interaction evenness quantifies how balanced the distribution is of interactions across species. And so we see that this interaction evenness increases over time. And one might hypothesize that this has to do with, again, the evolution of species and how they become more closely co-evolved or adapted to one another. We also have generality and vulnerability. And the take-home message here is that species on older sites are more specialized. So Vulnerability increases over time. Vulnerability is defined as the average number of arthropods per plant. And generality also increases over time. This is the average number of plants per arthropod. So we also have nestedness. Nestedness essentially is getting at the way that interactions are organized. So with a nested network, the interactions are organized so that specialists are interacting with a subset of the species with whom generalists interact. And kind of the take-home message here, because we can see that network nestedness is decreasing over time, is that it's decreasing with site age, given the high probability that these newly arriving species at the younger site are interacting with generalists that are already present. Kind of the contrast of interaction nestedness is the organization of interactions into more compartmentalized organization, or what we call modularity. And so here we actually see the inverse pattern. We see modularity increases over time. So at the youngest site, there's lower modularity, and then there is at the oldest site. So this likely has to do with the fact that there's, again, more coevolution or specialization over time, and species that are interacting in more one-to-one relationships. So when we look at this ecosystem level of change over time, we see some very predictable changes. And a lot of these changes, again, are related to specialization over time, or the change in the relationship of the arthropods and their associated plants so that they are more tightly co-evolved. The last little bit, I want to explain to you how we can highlight and use this method to understand community stability. So if we think about a hypothetical community that's undisturbed, we have, predators, and we have actually top predators that may be feeding on other predators. Those predators are feeding on organisms that perhaps are parasites or are pollinators and have different roles in the community. And those then are acting on kind of the base of the food web. And when we have a disturbed network, in this case, the introduction of invasive species, that changes the way that these organisms are associated. So in this diagram, the weight of the interaction is the strength of the line. And you can see that the association is, negative versus positive. And so it really changes things. And so when we look at our network data, we can pull out some stories that tell us a little bit about how introduced species are changing the community stability. So in this case, I just looked at spiders, and we have this network of spiders on the top and their prey community on the bottom. Everything that's highlighted here are introduced spiders. So if they're highlighted in a color, that means that they did not evolve on Hawaii and they essentially don't belong there. And they're having these novel interactions with the prey that are there. They're taking away the prey from the native spiders and potentially taking away the prey from the birds as well, and so on. Another clear example of this is the ants in Hawaii. So this entire family is introduced on the islands. There aren't any native ants. But you see that at the younger community, these invasive ants species are in really high proportion. This thickness of this line compared to other Hymenoptera, and there are much fewer in the older islands. Kind of a similar story here, just thinking about at the youngest community to the oldest community with the Hemiptera. So these are our sap feeding insects, and they're really depending on Metrosideros polymorpha. At the older community, they've diversified onto different plants. So we could ask the question then, are older communities more resilient to these changes? Like if I remove Metrosideros polymorpha, at the older community, Hemiptera could just switch to a different plant. At the younger community, they would be in a little bit of trouble. So in conclusion, using these remote oceanic archipelagos and combining it with this DNA methodology, we can really capture the dynamics of biodiversity, and we can capture it at multiple different scales. What we see is that biodiversity, or the change in accumulation of species, seems to be related to how the ecosystem is developing or the island developing as well. And also that these kind of ecological processes of colonization, for example, seem to dominate in the younger community, and evolutionary ones become more important over time. And we also see that if we look at like the resilience of communities, that older communities may be more resilient and younger communities may be more likely to be invaded. And we know that these novel speciation or novel species interactions are driving a lot of change, and it can have really rapid effect on these network properties and on the community and the ecosystem as a whole. So with that, I need to thank the organizations that donated voucher specimens and the individuals that donated voucher specimens to help us build our DNA reference library. And we also did a lot of literature databasing to figure out what the role of these organisms were in the environment. There's a whole bunch of wonderful undergraduate student researchers that helps with this project over the time that I was a graduate student, sorting specimens, helping with literature review, etc. And also my lab group, which has just been a wonderful place for discussion and bouncing ideas around. And a bunch of funding sources and organizations and collaborators that have helped with this work. And with that, I think we have some time for questions.
Yeah, if anybody has any questions, feel free to put it in the chat. I did see one in the chat, but I think it was answered. If not, we can follow up on that. But yeah, feel free to unmute yourselves if you would like to ask any questions.
Don't be shy.
I'll give you a Luddite question here.
Okay.
So, you know, the relatedness increases over time, you know, the distance between the differences, I guess. But is that, I mean, arthropods obviously live in a sort of micro scale compared to larger animals. So how does island bounding affect arthropods with respect to that kind of thing? In other words, if you did this study in the continental United States where it wasn't so separate and you were looking at these different little villages, so to speak, would they have the same kind of connectedness, or is some of that related to the island bounding?
That's a great question, Angus. So part of the beauty of having the island system itself is that it's easy to separate out the role of colonization versus evolution in adding species. And so that's why we can really predictably see this change in the distance between relatives over time. I think that you wouldn't, if you, let's say that you just had another discrete system, but you didn't necessarily have the time element, you could still probably get at, you know, the differences between the communities. But as you pointed out, if you have populations that are what's called panmictic, meaning that they're just all kind of, there's a lot of gene flow between them and they're all kind of relating to one another, then in that case, it would be really difficult to discern. So part of it is the island effect. Part of it is even more amplified by the great isolation of Hawaii. So we separated the native and non-native taxa for that analysis. Actually, for all of these analyses, I separated non-native from native because it didn't make sense to talk about the evolution of species, but also have the influx of things that have been arriving over the last 100 years or moving around more rapidly. And so that would also kind of squash that signal of relatedness if you included the introduced taxa. So I think that in answer to your question, if one wanted to do this in the continental US, one would still want discrete communities that are somewhat bounded, and the ability to sort of separate out, you know, how taxa are evolving in that area versus being introduced from other areas. But otherwise, you should still be able to sort of pick up that pattern. And then again, that gets back to this sort of community assembly paradigm of like that local pool versus the regional pool. We know that species are moving back and forth between them. And then we also know, given enough time, they're going to evolve.
Cool. Great talk. It's really fascinating.
Thanks for coming.
Awesome. It's Susan, you have your hand raised.
Yeah, hi. Great presentation and great research, Heather. Really enjoyed it. And you did a great job kind of breaking everything down. So appreciate that as well. I just had a collection question about if time of year matters when you're doing your collections. And if so, how might you expect that to change? You know, we have sort of two very strong seasons here in Hawaii, and I'm just wondering how that might impact your data collections.
Yeah, that's a great question. So with the very wet, rainy season versus the like more dry season and seasonality with arthropods in general, we know that that they will emerge and go through life stages, and then also kind of go into a quieter period as well. And what we did was we tried to just sample in the wet forest at these like discrete, within a certain elevation precipitation band. So that kept us in the wet forest, which kept some fluctuations down. And we did mostly sample in the summertime, in the summer months. So we tried to, kind of keep it within the period when we know that most of the arthropods have like emerged and are going through their life stage cycle. But absolutely, it would affect your ability to like detect things a little bit less so because you're using DNA metabarcoding. So maybe you're still picking up the larval form, even though you're not sampling the flying form. And also because we did this like kind of comprehensive sampling across all of the stratified layers of the forest. So if something had changed from like larval form to flying form, we would pick it up in the malaise trap. But yeah, so it's less like of a factor than it would be in more of like a temperate environment, for example, where things have gone completely dormant. But yeah, it definitely can have an effect.
Great, thank you so much.
Awesome. Thanks, Natalie, for speaking today. I will be sending a follow-up email with the feedback form and a way to contact Natalie if you have any more questions. Thank you all for coming today. Natalie, we can stay on the call.